In terms of viral vector vaccines, the virus, we can use a live virus, a genetically modified live virus, to carry the DNA into human cells, and this DNA contain the virus encodes antigens, which is once expressed in the infected human cells, it can elicit an immune response, which means virus can serve as a delivery platform to deliver the DNA vaccines, and in which this vaccine, the DNA, is encoded for specific protein antigen. So viral vectors combine many positive qualities of DNA vaccine with those of live attenuated vaccines. So like I mentioned in our previous lecture on recombinant vaccines, when we use a subunit, for example like protein or just DNA, it lacks the danger signals compared to the live attenuated vaccines. So when we use a viral based vector, we can somehow emulate the danger signals that the live attenuated vaccines possess. But instead of delivering the genetic material of the virus, we can modify it with the gene that encode for specific antigens, maybe the protein antigen. So most of the vaccine delivery utilize adenovirus as a vector. They use an adenovirus which has been engineered to be incapable of replicating and causing disease to deliver the genes for making the specific antigen. In case of viral vector-based COVID-19 vaccine, the gene that they carry is the spike protein of the SARS-CoV-2, the SARS-CoV-2 spike protein. So adenovirus are so common to human, which create one problem with using them in vaccine is that people may have antibodies to them. So researchers get around that issue by using adenovirus that humans are unlikely to have encountered before. For example here, the AstraZeneca and Oxford COVID-19 vaccines use a modified version of a chimpanzee adenovirus. Another issue is that we know that adenovirus deliver DNA that can enter the cell nucleus, which brings up the questions of whether they can alter DNA. Easy answer is no. They cannot change the DNA. Adenovirus, even as they occur in nature, just do not have the capacity to alter DNA. Unlike retrovirus such as HIV or lentivirus, wild-type adenovirus do not carry the enzymatic machinery necessary for integration into the host cell's DNA. That's exactly what makes them a good vaccine platform for infectious diseases. So this is just a diagram that to show the differentiation between the viral platform. For example, the virus with the replication defective vector, which is used for gene therapy, and replication competent vectors. Later we will see on how we can design the virus to act as a drug on its own for cancer oncolytic viral therapy. And then we have a virus that act as a delivery platform for antigen delivery in the form of vaccines. And then we can have a virus-like particles. This is similar to the one in the recombinant vaccines, which utilize the concept in the development of Gardasil, where they self-assemble the protein, that the capsid protein of the human papillomavirus, and they can self-assemble to form like a virus-like particles. This is different to the when we use the viral as a vector. So which means that this is a vaccine on its own. And then we have the inactivated or attenuated virus that can be used as a vaccines as well. Alright, so you need to be able to differentiate the different virus platform and how it is used for certain diseases or for certain therapy. Right? And next is we will see on how we can use viral vector for cancer immunotherapy, and it can be categorized into two, which is the direct or indirect gene therapy for cancer. For the direct gene therapy, it involves the insertion of gene to tumor cells for the direct killing or by suppressing the abnormal growth by gene products. The gene used for this strategy are suicide genes, functional tumor suppressor genes, antisense genes, known as oncogenes or anti-angiogenic genes. While for indirect gene therapy, it will involve the insertion of a gene that modifies or stimulates immunocytes to be more aggressive or effective against the tumor cells. Some types of the gene therapy also aim to boost the body's immune response to attack cancer cells. And this sometimes we can term it as a cancer vaccine. And cancer vaccine can also denoted to act as therapeutic cancer vaccines, in which, once stimulated, our immune systems has cells that can recognize and kill harmful things that can cause the disease, such as cancer cells, but it need to be triggered first.